Now we'll be walking through the steps to measure resistance with the Millicell ERS 3.0. To start, take your cell-seated insert plate out of the incubator and place it in the cell culture hood. Set your timer for 20 minutes to allow the plate to reach room temperature. This time can be spent cleaning your surfaces and your Millicell ERS instrument and probe. Spray the surface of the instrument with alcohol prior to placing it in a cell culture hood. Disinfect the electrode probe with 70% alcohol either by wiping the surface with a Kim wipe or submerging the legs of the probe in alcohol. Let the probe air dry in the hood and rinse off any residual alcohol with PBS. This will also help to reionize the surface of the electrodes. We'll go into more detail on this in Chapter 5. On the instrument's touchscreen display, first select Plate Map for use. Then select Add Data. Users have the option to navigate their plate in the order of their choosing. We offer the capability to rearrange the order by group, by row, by column, and a variety of snaking patterns. Read an entire plate in under 10 minutes. The standard electrode probe fits 6-well, 12-well, and 24-well plates. To ensure optimal results, rest the adjustable ring flat on top of the well plate so that the sensors are submerged to a consistent depth in the wells. Avoid crooked placement of the probe, as this can lead to inaccurate results. For the most accurate readings, adjust the ring so that the gold sensors are completely submerged in your media. Please note this example of incorrect placement. In this instance, the electrodes are set too high. This will result in unstable readings on the instrument. To resolve this issue, simply turn the ring until it is closer to the probe body. This lengthens the electrode legs and increases their depth in the well. Adjust the ring to the correct height for your culture wear. This can vary, but always make sure the gold electrodes are submerged in your media and that they do not touch your cell membrane. Note the correct placement and the electrode submersion and how the probe sits flat on the plate. If it is necessary to wash the electrodes between measurements to avoid carryover of one sample into the next, the electrodes should be rinsed with the experimental culture media or PBS. It is best not to rinse the electrodes with Millicell water or distilled water. Doing so would change the ion surface chemistry on the electrodes of the probe, which can cause drift in your measurement. If this incident occurs, place the probe legs in PBS for several minutes to recover from exposure. The measurement screen shows the plate layout, the plate name, the user progress for measuring wells, buffer average if applicable. The time since measurement started is located on the side. The well reading is the background subtracted value in real time. But don't worry, the raw value is also displayed just below it next to the temperature reading. To record a measurement, select Save and Next. The instrument highlights the well that it thinks is actively being read in a red circle on the plate map. To change the active well to the desired well, tap on the plate map and simply tap on the well of interest. Tapping on the plate map will expand the plate into full screen view. To change the active well to the desired well, select on the well of interest. To return to the measurement screen, either select a new well on the plate map or select Cancel. When finished taking measurements, select Finish. You'll then see a summary of measurements with toggle buttons for each measurement type: resistance, voltage, and temperature. If the measurements are completed, select Save to save the data to the device and Millicell Cloud account. To return to the measurement screen, select the back arrow on the top left of the screen. To restart measurements in voltage mode, select the Add Voltage Reads button. View your data in the Millicell Cloud and download raw files to your computer. Log in to the Millicell Cloud to view your data and projects. All measurements are present on the cloud from Millicell devices, such as DCI and ERS. Data can be easily downloaded as a PDF or CSV file, opened on your computer, and analyzed. For more about the cloud, please tune in to our next video. For more information, please contact your account manager or visit our website.